Siglec-6 is a novel target for CAR T cell therapy in acute myeloid leukemia.
I'm Kevin.
I'm Isabel.
I'm Coven.
And I'm Sienna.
Okay, so AML or acute myeloid leukemia is essentially a type of blood cancer that originates in bone marrow, and bone marrow is where blood cells are made. And so it's characterized by these like rapid growth of abnormal white blood cells that like build up in the bone marrow and then interfere with the production of like normal blood cells, right? So hematopoietic stem cells are like where it originates, and basically they can have the potential to like turn into all these different types of blood cells. And so through like genetic and molecular changes, essentially what you can have is like clonal expansion, which is where this like mutated like cancer cell takes like this growth advantage over the other cells and then starts multiplying and everything. This is like a significant like characteristic of cancer, right? And so they often lead to like these like differentiation blocks, and so they prevent normal blood cells from like, or like normal like maturing into like blood cells, and essentially they can lead to things like anemia, neutropenia, these different types of blood illnesses.
Yeah.
And so CAR T cell therapy is basically like collecting these like T cells, these white blood cells, and then basically re-engineering them in like a lab to produce proteins on their surface, which are the CARs, and these like bind and recognize specific proteins or antigens on the surface of these cancer cells.
Okay, yeah. So what are CAR T cells and what are their structure? So this is the basic structure of a CAR T cell. So you have your antigen binding domain, which is the main thing that they change in the lab to bind to the various receptors that we have on our cells. A hinge domain to connect it to the transmembrane domain, to connect it to the intracellular signaling domain. So when something binds to the upper antigen binding domain, it can lead to a signal transduction pathway and activate certain things later down the chain. And we CAR T cell possibilities. So CAR T cells, we've glossed over it mostly, but it's not just engineering a certain protein on a T cell just to target one thing on a cancer cell. It has these possibilities that we can talk about. So there's this Boolean logic. I don't know if you guys have taken computer science. You have like or,
Oh
like
yeah, yeah.
or and these Boolean functions. So what you can actually do to make CAR T cells more specific is that instead of just targeting one, like targeting Siglec-6, you can target Siglec-6 and then another cancer factor on AML to make it even more specific to make sure that it doesn't affect other cells in your immune system. So that's what this is up here. Down here, it basically showcases the different CDs, or CD is just another name for a protein like a receptor on our cell surface. Siglec-6 is just another name for CD327. So you can target all these other CDs using CAR T cell therapy and treat all of this variety of diseases. So B cell leukemia, AML, HL, MM, and so on.
Hmm.
On the top right as well.
Okay, so the top right is basically how we can use CAR T cell as like an anti-anti. So something that tumor cells produce is this receptor that binds to the anti-PD-L1. L1. And what this does is if it binds to like a T cell, it'll like render it useless. So dispose of it, right? But what these CAR T cells can do is have something that is essentially blocking the anti-PD-1, right? So this makes it so it's a natural defense, not a natural defense. It's just a defense mechanism against these tumor cells like attack.
Yeah. So we've all heard of like relapse in cancer. So these cancer cells develop resistance or like somehow inactivate your immune system. And CAR T cells have the possibility to overcome that.
Like that's a big thing in AML. Like from what I'm doing research on like AML, right? And so this big thing about it is one is like resistance, like chemotherapy, things like that. But another is like cancer remission or coming back after a certain period of time. So it's interesting to see how like CAR T cells can actually prevent that in the future.
Yeah. And then the last one is just right now we focus mainly on CAR T cells to like directly attack leukemic cells. But in the future, we have the potential to engineer CAR natural killer cells, CAR macrophages, or even CAR helper T cells, like memory. And it overall just expands what we can do with genetic engineering to help healthcare. So what they actually did in the study, this is a visual abstract. They used genetic engineering, like CRISPR, to design their Siglec-6 CAR T cell, and then they expressed it in T cells extracted from mice to produce these CAR T cells. And they put these CAR T cells back in the mouse. And you can see here, oh, okay, cytokines and cytotoxic granules. These are cytokines. Basically, higher levels of them means a better immune response. So as you can see, higher levels of them attacking AML blasts and CLL B cells, wiping out the leukemia. And we'll get more into this list later, but specificity, it's not attacking regular hematopoietic stem cells or memory B cells.
And another interesting thing that we'll also touch more on later is like you can see here that the genetically engineered part that we're adding on is actually derived from like the human JML1, so it's naturally like derived, which is always better.
Okay, so the first thing that they did in the study was validate Siglec-6 expression in AML cells. So how they went about this, figure A, they used a process called flow cytometry. So they basically cultured these cells, used a micropipette to put them on a plate, put them under a microscope, and then this computer program just counted them for them. So in this graph, the further you go to the right, the higher levels of the thing there is. So the red is the Siglec-6 and the blue is an isotype control, a control that's present in every cell and at the same level so they can compare back to it. And basically this, you can see this red peak is farther along than the blue peak. So in this U937 cell, there were higher levels of Siglec-6 than the isotype, meaning it's like a pretty strong leukemic cell. And other cells that they use in the study, MV411, MOLM13, Kasumi1, I think that was a control cell, but MOLM13 and MV411, they were also leukemic cells, but expressing Siglec-6 at a lower level.
And then the other figures are basically showing the same thing, that they're validating like Siglec-6 in these AML lines. So C is showing the mRNA transcript. So again, we can see in U937, MV411, MOLM13, there is Siglec-6 being expressed to varying degrees. And then, of course, sometimes like mRNA won't always be transcribed. So the next thing that they did is counting the molecules of Siglec-6 to really show that, yes, like this is being expressed and not just mRNA is being generated. So again, figure D is showing that the U39, MV411, MOLM13, all these AML cell lines, like Siglec-6 is being expressed. So.
Okay, yeah, and then did you talk about F already? Okay, so basically in F, it's basically showing when there's like the IFN gamma and the interleukin, which are both cytokines. And basically in this, what we're looking for is like a higher level, which means that essentially means the immune system is working better, right?
Yeah, I think it's really significant that they had two different versions of the Siglec-6 CAR T cell, the blue and the green, but they also tested UTD T cells, just natural T cells.
Oh yeah.
And it's important to note when UTD T cells were infused into these,
it's not as effective.
Like the cytokine levels were not at all, meaning they were not working well.
So just naturally it doesn't like work well. So that's why you need like so naturally these like T cells in the body don't work well. So do you use the Siglec-6 to like supplement it, or is it more so like screw the T cells?
I think it's just like a direct replacement.
Okay, so screw the T cells, the Siglec-6 is taking over.
Yeah, yeah, basically. But I think it's important to note that like they used flow cytometry, like qPCR, counting proteins. They used all these methods, and it just highlights how thorough you have to be in your biological research to actually validate your findings, because you don't want to like associate this like Siglec-6 with AML maybe there's a confounding factor.
Okay.
Yeah. Like you might look at these and think, oh, like this is really redundant, but turns out it's not.
Yeah.
Okay, in this slide, it took 3 patients from the study, patient 10, patient 5, patient 1. Patient 10 was the most sick with leukemia, patient 5 was more the middle, patient 1 was the least sick. And it shows in part B just the contrast between like the natural T cells, which is the blue, and then the Siglec-6, and how effective the latter was, because you can see in all 3 patients, more so in the more sick ones, that the Siglec-6 like had 100%, near 100% cell lysis at varying concentrations. So even at a lower concentration of the Siglec-6, it's still effective. Contrasted with the natural T cells, like with the more sick individuals, like it was almost like 0 for patient 10 and then around 20% for patient 5, which means that like cell lysis, like breaking down the tumor cells, it was very ineffective, and then the patient would inevitably like pass.
Yeah.
Mm.
Another thing that's important to note was the E to T ratio, which is the x-axis on all of these graphs. E to T is like effector to target ratio. Think of it as like effector, your killer cells, and then target your leukemia cells. If you have more target cells versus leukemic cells, of course you would expect it to work well. But to screen for efficacy of the actual CAR T cell, the lower your ratio is of the CAR T cell versus leukemic cell, if it's still working well, then it means your CAR T cell is specific, it's efficient at killing these cells.
Yeah.
So like you can see 10 to 1, 5 to 1, 2.5 to 1 ratio, the smallest ratio. All of these Siglec-6 CAR T cells were still like reasonably effective against these.
Significantly effective.
Yeah.
And like, for example, in patient 1 who was not very leukemic, like sure they're UTD, which is like they're untransduced, so they're, they're natural T cells. In the LSC one, in a 10 to 1 ratio, so 10 UTD cells to 1 cancer cell, like it was pretty effective, around 50% cell lysis. But as like the concentration drops, so only having 2.5 UTD cells compared to 1 leukemia cells, like it's 0. It can't do much. While Siglec-6 is still at like 80%, 90%.
Mm.
Right. Next is
This
just
slide
further
was kind of-
validating again the in vivo effectiveness of Siglec-6 CAR T cells. Kevin, you want to go with this?
Yeah. So what I found really okay. So this is basically just recapitulating kind of what's been shown in the other slides. You can see the high level T cells or UTD T cells, Siglec-6 are both forms of Siglec-6.
Oh, sorry.
And then in like E and F, you can see bone marrow, spleen, peripheral blood. But what I found really interesting was in F, how on day was like between 14 and 21, just it was a significant drop from like 100% survival to like 0% survival, like for the UTD T cells. And
Yeah.
this was like because like I think the mice were like experiencing too much pain, and so they had to euthanize
Like
them.
it just wasn't working enough.
Yeah.
They just had to kill the mice to
Yeah.
Yeah.
Because basically in this one, they took like, I think it was like 24 mice, divided them. Oh, these ones get UTD, these ones get Siglec-6 28Z, these ones get Siglec-6 BBZ. And okay, let's see how they fare over time.
Mm.
Did not take
They
long.
did not
the
fare well.
natural T cells did not do well.
But you can see the Siglec cells, the overall survival just remains at like 100%, which
Yeah.
just
For
shows
both
how effective. Yeah, for
variations,
both
yeah.
variations, which actually just shows how much more effective it is, or just further emphasizing the effectiveness of it compared to maybe what your body would regularly produce to combat, AML.
Mhm.
Also,
And I
another
think,
thing, like when you're reading these studies and then the figures are just so confusing, like it takes you a while to take them in. Like these figures are so obvious that the Siglec-6 is working well. The red,
Oh
Yeah.
yeah.
the higher the red is, the more leukemia cells there are. So the red is the UTD or the natural T cells, and they're just the leukemia cells are still there. And then the blue and the green, the Siglec-6, they're just gone. So it just
Like-
wiped out the leukemia.
Yeah, like especially like the bone marrow, that's where like it's all produced, right? And you can see that with the Siglec-6, both variants, it's just 0%. Like at the center where it's produced, there's just no leukemic cells whatsoever, which I thought
Mhm.
was really cool.
And also I think, of course, like the two Siglecs, they had different effectiveness. Like I'm pretty sure BBZ was just a little bit better, but interestingly, both of them after enough time, like they were like the mice were like completely out of remission, like no developing cancer again for both of them, which is incredible, obviously.
Oh yeah.
And it's effective throughout the body, like
Right,
like
yeah.
Oh
also
yeah.
Bone marrow,
Not only
spleen,
the
blood,
bone marrow, spleen, and blood. Yeah.
like all the blood circulation.
Yeah, I don't know if I explained that earlier, but like AML, it starts to affect the blood processes like all throughout the body, which then begins to affect like other functions, not just just blood circulation.
Right.
The blood does a lot, so that's
Mhm.
Yeah.
very
Yeah.
good.
Like blood is how cancer metastasizes, like how
Yeah,
it
yeah.
spreads
Yeah.
to different tissue. The next thing that they did was actually validate the specificity of Siglec-6 CAR T cells. One major problem with current therapies, or like any therapy in general, is how specific it is. Like people with cancer get treated with chemotherapy, but like, is that really effective? They're just killing everything and hoping,
Mhm.
like
Oh yeah.
hoping that the cancer dies before you die. You know, that type. So,
And it has a
um,
lot of other side effects,
Oh
right,
yeah.
yeah.
fatigue and vomiting and
So
nausea. And
so it's really important to have. So next, they actually validated the specificity of the Siglec-6 CAR T cell. So in Figure C, they did a PCR analysis on the Siglec-6 mRNA transcript, like before. So patient 24 was highly leukemic, and MOLM13 is an AML cell line. So they have, of course, higher levels of Siglec-6, but Siglec-6 was not expressed on HSC, hematopoietic stem cells, or just natural cells, and Kasumi1 control. So it just shows how Siglec-6 is a target on these leukemic cells. But if you have that CAR T cell that's screening for that Siglec-6, it won't identify these natural cells that you want to keep.
Okay, yeah. So it's
Yeah.
just not affecting anything that it doesn't need to affect.
Right. Yeah.
Okay,
And
okay.
then D just further validates that donor 1, 2, 3, 4, 5 are all healthy patients. And this is a flow cytometry analysis, same as before. So Siglec-6 levels are the same as any other like control isotype in the body. So it's just not higher levels of Siglec-6 and
Okay.
normal expression.
And this like relates back to what I think Kevin said earlier. So obviously this is like novel and really good because unlike chemotherapy or radiology, where they're just like, throwing in these chemicals and hoping that it'll kill the cancer while it also kills everything else, right? This is a lot better because it's specific and targeting only the cancer and not like your other healthy cells.
Right.
Mhm.
This final figure here, Siglec-6 CAR T cells. So they use these CAR T cells to target not only, we covered this in the visual abstract, but not only AML, but also BCLL or B cell chronic lymphoblastic leukemia. So this can target two different types of leukemia. And again, Siglec-6 higher expression as you go towards the right on the on the x-axis leads to higher specific lysis or destruction of these leukemic cells.
The more
Just further
Siglec-6 you
validates.
have, the more
Like
specific
higher
it is.
concentration.
Right,
Okay,
exactly.
okay.
And finally, conclusion. So
Okay.
basically, in the study, just Siglec-6 is this novel target antigen for CAR T cells in AML and BCLL.
It's also important to note that there were, I mean, this is obviously Siglec-6, so there were 1 through 5. For 2 and 3, they were notably not as specific, and it might have like killed the normal like blood cells in hematopoiesis, like processes that would like have a similar effect as chemotherapy and radiation, which is bad. But Siglec-6 is extremely specific, as we talked about in those past 2 slides, and that's a very promising way like to target cancer because it doesn't affect these normal cell processes, so you can recover faster and it's more efficient and healthier.
Yeah, it's also important to note that like we're talking about all these like, oh, this is so novel, it's so specific, it's so effective. And maybe you're wondering like, okay, why aren't they just using this on like patients with leukemia now? It's because these research is just the first step in like a drug discovery journey. It has to go through years of clinical trials, and maybe down the line it'll be screened, and then maybe it just causes like some other cancer and it's just not effective at all. So it has to go through these trials to make sure they're actually safe in humans. So like this is just a preliminary trial. Like they're testing leukemia in mice. This won't come to the market for another 10 years, and that's if it survives the trials.
Mhm.
So
But
that's
for
just
now, it's very promising.
yeah,
Yeah.
right.
That's exciting.
And another really cool part of Siglec-6 is that it's derived from a natural monoclonal antibody. I think it was actually also in the visual abstract JML1.
Mhm. Yeah, that's obviously good because let's say you're doing things like donating organs or something. Oftentimes, you have things like organ donors, right? Their organs might get rejected from like the host body, or things like blood donations where not everything might be compatible with each other due to kind of various sources, but it's good to have naturally derived because it basically improves compatibility, right? You don't have to stay in the organ donor list and wait for like a new organ transplant where people are normally like, it takes years for them to get, you know, their desired organ that like fits their body and everything. And the unfortunate truth is that a lot of people don't get what they need in terms of blood donations, stem cell donations, organ donations, and they end up passing away. So it's good to see something that's actually naturally derived from one's own body.
Yeah. But I think it's also cool to note that of course it's important, like having a medicine that's naturally derived is incredibly useful. But I don't know if y'all have heard of like they have like synthetic organs, or they're trying to
Ooh,
develop
yeah,
those
yeah, yeah.
now as well. And I think as science improves and we're able to mimic these biological processes more, being naturally derived is not going to hold as much significance.
Mm.
Yeah. But for now, it's very nice because it stops these adverse reactions and stuff.
Right.
For now.
Cancer is bad. Yes.
I mean, it gives you the template already. You're making a like CAR T cell, like you're changing the genetic code, but you already have that baseline. It's like, oh yeah, it's not going to get rejected as long as you don't change too much about
Yeah.
Because it's from yourself. Like these CAR T cell therapies, they take T cells from the patient themselves, genetically engineer it, put it back in them. Right.
Mhm.
So
Right.
okay, that's a wrap.
Anyways, thank you guys so much for
That's a wrap.
watching. Don't forget to subscribe,
Okay.
like the video.
Like,
Huh?
comment, subscribe, share, ring the notification icon.